Good morning, everybody. I am Narayan Lamichhane from University, Assam, and I am from Nepal, and currently I am studying in PhD program in University. Today I am going to talk about the biosynthesis of spectinomycins, which is also a kind of aminoglycoside antibiotic produced from Streptomyces spectabilis, and the characterization of the biosynthetic genes of spectinomycins, and after the elucidation of the genes responsible for the formation of spectinomycins, which can give the information for the production of different kinds of hybrid antibiotics, which are the new and global potent activity. This is spectinomycins, and structurally it is a simple aminoglycoside antibiotic, and it contains the two moieties. One is this actinamide moieties, and another is spectilose moieties, and these are joined by the aminoglycoside bonds. And actually, these actinamide moiety play the important role in the binding to some ribosomal parts of the microorganism, especially this NS and OS group make the hydrogen bonds with these regions of the cytinous rRNA of the Neisseria gonorrhea, and this spectinomycin is clinically used in the treatment of gonorrhea. This gonorrhea is caused by the microorganism Neisseria gonorrhea, and so it makes the hydrogen bonds by this NS and OS group to this position, and it blocks the translocation state by combining making hydrogen bonds with the elongation factor G, and so the protein synthesis is blocked, and by this way it used in the clinical importance. Now I am going to focus about some characterization of the biosynthetic pathway of the spectinomycins and the construction of the gene cassette for in vivo expression. For the fully characterize all the biosynthetic gene of spectinomycin, we constructed one expression plasmid which can be used for the gene expression in the actinomycins. So for the in vivo expression, we constructed some gene cassette which can be used for the further characterization of the biosynthetic gene of spectinomycins, and which can lead the way for the production of hybrid antibiotics. And this time we are making some construction plasmid by the use of the biosynthetic gene of spectinomycin, and by this way later we can get some type of the new different types of substituted spectinomycin. It is the biosynthetic gene cluster of spectinomycin, and this slide shows the location of the different biosynthetic genes in the biosynthetic gene cluster. And out of the cellular ORFs, now we are working on these such type of the ORFs which are shown in the blue color, and these are some crucial genes which are found to be involved in the biosynthesis of spectinomycin also. Later I will discuss about this. It is the biosynthetic pathway of the actinamide. This is the actinamide plays the important role in the binding to some part of the microorganism, ribosomal part of the microorganism, and its biosynthetic pathway starts from the myeloinositol phosphatase, and then it is converted to myeloinositol, and it's from this myeloinositol it shows the 6-step process. First dehydrogenation, later transamination, and the same process of the dehydrogenation and transamination, and later it is the methylation, and at last we got the actinamide. It is the purpose biosynthetic pathway. Similarly, for the case of another moiety, it starts from the TDP glucose, and from this, by the help of these enzymes, the last product is the spectilose, which is also purpose biosynthetic pathway, and this actinamide and the spectilose, these are joined by the glycoside transferase, we got the spectinomycin. And this time we try for the in vitro expression in the E. coli from the Streptomyces spectabilis. For this, we took some crucial genes, which is for SPCB myeloinositol dehydrogenase, and another SPCS which is sideroinositol aminotransferase. And these are the expressed under this condition, and this enzyme also expressed under this condition, and the enzyme is carried out. And in the case of dehydrogenase reaction, as we see, this OH group is converted into the ketone, and it is NAD plus dependent myoinositol dehydrogenase, and the conversion of the ketone can be easily detected by the C13 NMR. And we took the C13 NMR, and the conversion of the ketone is detected at 180 ppm, and which was confirmed by comparing with the myoinositol dehydrogenase, which was purchased from the sigma, which has taken the reference. And it is for the enzyme SPCS2, it is the amino transferase. So the conversion of the amino group can be easily detected by the spraying with the neohydrin reagent to the assay product, and by the help of neohydrin reagent, if the product is converted into amino form, it will get the this type of the red color, and it is also detected by the LC-MS analysis. So the product was found at 180 mass. It is for the another enzyme, this is the N-methyl transferase, and for the case of N-methyl transferase, it is the reaction in the 2-deoxystreptamine, and it is so the conversion into the this amino groups will convert into the this methylated form. So the N-methylated deoxystreptamine, and we got the protein under this condition, and we check by the TLC, and from this TLC, the sports will be on the expected sign for further characterization of this enzyme is going on. These are the methods from which, by the use of actinamide, we can make the different types of hybrid aminoglycoside. So I mentioned that the SPCB, SPCS2, and SPC8 genes are involved in the biosynthesis of the actinamide. So now we are in the process of the in vivo expression of the actinamide gene, and after the in vivo expression, we can insert the genes into the butyrosine as well as in kanamycin, so that we can get such type of the hybrid antibiotic. For this, we can knock out this deoxyribonucleic from this kanamycin, butyrosine for other aminoglycoside antibiotics, and we can use this actinamide, which is found in the spectinomycin. The actinamide and the deoxystreptamine are very similar. It is different only in two positions, C2 position. So we can insert this P-actinamide into this deoxyribonucleic gene. We can disrupt, and we can get such type of the butyrosine. And it is also same type of strategy for the production of the hybrid gentamicin, and it is modified streptomycin. And for this purpose, we clone these genes responsible for the actinamide. And to clone the actinamide gene, we constructed one vector. This is high-level expression vector in the actinomyces, streptomyces. We constructed namely the PBR100 and PBR20. It contains erythromycin promoter. It contains the ACT promoter, which is obtained from the streptomyces CD color. It is the ACT second, where we pass this to regulator gene, and these three genes are cloned, and these are the expressed under the control of this actinorhodin bidirectional actinorhodin promoter. Similarly, these are cloned, and it is controlled by the erythromycin promoter, and by this plasmid we can get this such actinamide. So for this purpose, I am going to describe how we constructed this high-level expression plasmid to produce the different types of enzyme in the high-level expression. It is the expression plasmid we constructed, which contains the pathway-specific regulator gene bidirectional promoter along with the multiple cloning site. It contains the histidine tag fusing, so that the protein after expression can be purified by the nickel affinity chromatography. And this is the size of the plasmid vector we constructed by this method. And I will go which method we designed such that the plasmid is the first we amplified this bidirectional promoter, and we clone into the PLJ plasmid and construct the IBR10, and later it is converted into clone into PGM7 PGM7 vector, and after cloning the multiple cloning site along with the histidine tag, we constructed this PIBR100 having the size 13 kb, and similarly it is also cloned into PWSN3, and we modify the PWSN3 along with this actinorhodin promoter, and we are named this PIBR150 having the size 8.5 kb. And these are the some DNA manipulation method for the construction of this plasmid. It is through amplification of the multiple cloning site from the PRACTV, and it is the amplification of the bidirectional actinoleurin promoter, and later we clone in the PSGF plasmid, and it is transferred and it is converted into PIBR10, and later this PIBR10 is modified after cloning the multiple cloning site. And this is the multiple cloning site, and it is the PIBR10 containing the actinoleurin promoter and this pathway specific regulator gene. And after this cloning, it is again transformed into the clone into the PSGF plasmid, and at last we got the PIBR100. Similarly, we cloned this multiple cloning site into the PWSN3, and we got the PIBR150 having the size about 8.5 kb. So by this way, we constructed these two plasmid, and we cloned the biosynthetic genes of actinamide. Now we are in the process of transforming these actinamide biosynthetic genes into the hybrid kanamycin biosynthetic gene as well as biosynthetic biosynthetic gene for the production of modified aminoglycoside. This is the method of another deamidinated streptomycin production. This is the streptomycin produced from the Streptomyces graeseus. This return is amidinated, and it is converted by the STRB1 gene, which is called amidinotransferase. And we disrupted this gene, and after disruption, our target is the production of the only deamidinated, only containing NH2 group. And later we will check the activity, and this time I am going to tell about how we constructed this plasmid by the DNA manipulation method. DNA manipulation is the PCR amplification, cloning, and after this is the EDC method and converted into PKC1 and 3 vector. Now we are in the process of transformation into the Streptomyces graeseus after this disruption plasmid. And similarly, in another case, we are processing the production of hybrid, Streptomycin by the disruption of this young methyl transferase gene. In this case, we this STRB1 gene from the Streptomyces graeseus. This was taken by the PCR, from the PCR it was taken, and it is disrupted from this to this portion, this SPCH, it is N-methyl transferase of the spectinomycin. In the spectinomycin gene cluster, we introduce this gene from the Streptomyces graeseus, and by this way, in our expected product will be the, instead of methylated group, we will obtain the amidinylated spectinomycin. We contain the NSC, NS2, this type of group, and we can expect that its activity will be higher than the spectinomycin, because in case of spectinomycin, it has been proved by the crystal structure in another method that the hydrogen bonds will be formed by this NH and OH group with the ribosomal part. So if we added these groups, then better hydrogen bond, more hydrogen bonds will be formed by these groups to the ribosomal RNA of the microorganism. So it should show the better activity than the previous one. It is the DNA manipulation method, and use the viscous meat. It is taken into cloning PDM7 in insertion of the D gene from the graeseus, and this construction of this plasmid. And now we are also in the process of transformation this plasmid also. In conclusion, we characterize these three genes which are crucial for the formation of actinamycin. From the actinamycin, this is dehydrogenase. This is transformation for the transmission aminotransferase. This is the methyl transferase gene by in vitro expression. And we constructed the actinamycin expression plasmid having the strong promoter, and which can use for the expression of the genes in the actinamycin for in vivo expression. And we constructed a different disruption plasmid having the genes of the same antibiotics or from the different organism, and we are in the process of production of different hybrid aminoglycoside, and now we are in the process of transformation throughout this case. And our further works is related with the construction of the gene cascade of all spectinomycin gene, and by this way we can overproduce the spectinomycin. It is necessary for the overproduction of spectinomycin, and from this vector which we constructed, we can clone the different kinds of genes of another organism also. By this way we can overproduce the antibiotic. into the production of the hybrid antibiotic by using the actinamide zinc acid. We can insert the actinamide zinc acid to the moiety of the different aminoglycoside, whose moiety is very similar to the actinamide moiety, and we can produce way and this production of these two sugars are spectinoed by the disruption of specific gene. Yeah, that's all.